Yes, it's the guy. In this video, we are going to talk about the protein synthesis in antibiotics. We have been talking about this in many different videos, but in this video, I want you guys to really visualize and appreciate how these drugs really inhibit the process of the protein synthesis. We have been talking about the A sites, the P sites, the E sites. What are these sites? So please don't forget to like the video, subscribe, and comment. Here, I have some examples of the drugs which belong to this class, and I have an illustration which I'm going to use to describe briefly about the protein synthesis process. Let's begin. After messenger RNA being transcribed from the DNA, it comes and binds to the ribosome, other ribosomal RNA. This is ribosomal RNA. But this is the ribosomal RNA for the 30S subunit. And also this one is ribosomal RNA for the larger subunit. This is the 50S subunit, and this is the 30S subunit. This is our messenger RNA. The messenger RNA uses the ribosomal binding site to bind to the ribosomal RNA. This is the ribosomal binding site. Ribosomal binding site, also known as the chain diagonal sequence. Chain diagonal sequence. When it binds to the ribosome at this site, means that it is fixed. Now, what is going to take place is the translation process to transfer the information coded from the DNA into the protein. At the ribosome, we have different sites. We have this site, it's called the E site. This is E site, this is P site, and this is the A site. We are going to see what they mean. The E site is known as the exit site. The P site is known as the peptidyl transfer RNA binding site. And for the A site is known as the amino acyl transferase transfer RNA binding site. Is the amino acyl transfer RNA binding site. So these sites, we are going to talk about them. What are they and how do they help in the protein synthesis? Now, when the messenger RNA reaches the ribosome, the first amino acid carried by the transfer RNA in bacteria, it's known as the formylomethionine. It's carried by the transfer RNA to the P site. This transfer RNA, we use its anticodon to bind to the AUG. This is the AUG, and this AUG is the codon, and it's the P site. So when it comes, to be like this. To use its anticodons to bind to this region, having the first amino acid. When this binds here, then the second amino acid carried by the second transfer RNA will also bind to the A site, but this one will depend on the messenger RNA sequence. So, meaning that when this one comes to the A site, we shall have this. So this is just a bond. Then another amino acid, also on the transfer RNA, we shall have this. Okay, let me use these different colors such that we can separate them. This is the messenger RNA. This one is having also the amino acid. And this one is having the first amino acid. So the peptidyltransferase activity from the ribosomal RNA, this is the 23 ribosomal RNA. This one is 16 ribosomal RNA. 16S, this is 23S. It is 23S ribosomal RNA. This is 16S ribosomal RNA. This one. So the peptidyltransferase activity from the ribosomal RNA, which is the 23, will help in the formation of bonds between these amino acids. When we form the bonds, we shall have the transfer of the amino acid from the P site from this transfer RNA to this transfer RNA. Mean that we shall have this. We shall have this. Then we have the first amino acid, and also we are having the second amino acid. And this transfer RNA is left alone. There's no amino acid here. This one has no amino acid in the P site. After this, the messenger RNA will be moved from 5 to 3 direction. We are translating from 3' to 5' direction. But the messenger RNA will move from the 5 to 3' direction, meaning that when you move the messenger RNA, this empty transfer RNA will come to the E site. This is the E site. Also known as the exit site. Therefore, this transfer RNA having the growing polypeptide chain will be at the P site. So we shall have this with the first amino acid, and again with the second amino acid. Therefore, the A site is left empty, meaning that it can accept any new coming amino acid on the transfer RNA. Therefore, we shall have the coming of this transfer RNA bind to the A site, which is empty now. We shall have this one coming. Therefore, we shall have this with the transfer with the amino acid. Again, peptidyl activity from the 23S ribosomal RNA, we shall have bond formation between these amino acids. At the end, we shall have this. This one will be carried from the transfer RNA at the A site, at the P site, the transfer RNA at the A site. So we shall leave this one, the transfer RNA, empty, meaning that we shall form this. And this. So the chain is growing. This transfer RNA is left empty. And we have this one at the E site. At the E site, we are having the empty transfer RNA again. But this chain is growing. Again, the messenger RNA will be moved from the 5' direction to the 3' direction, meaning that this transfer RNA will leave the ribosome. And the time will reach, even this transfer RNA also exit the ribosome, meaning that we shall have again the A site empty, accepting the new amino acid, meaning that the chain will grow until we reach the the termination codon. These are termination codon. The AUG, the UAA, the UAG, and UGA. Any of these. If these ones are reached, when they reach the A site, there is no transfer RNA which can recognize these codons. So when we reach these codons, the release factors will bind to these codons. When the release factors, they are proteins. We shall have proteins binding to this region when it reaches the A site. When they bind to this region, the release factors, these are proteins. When they bind to this region, when it's at the A site, we shall have the hydrolysis of the bond of the grown polypeptide chain. This chain will be hydrolyzed. So this chain will be here at the P site, but it will be released and then it falls into a protein, meaning that we have formed the proteins. And the process will continue like that. So we shall form another polypeptide chain in the same way when this one is done. So that's the whole process, how it moves. Therefore, here, these are some of the classes of the drugs. These aminoglycosides, they bind to the ribosome. They are protein synthesis inhibitors. They bind at the small s subunit, which is the 30s. But they bind to this ribosomal RNA, which is the 16s ribosomal RNA. At the 16s ribosomal RNA. Mean that they can inhibit the process of they can inhibit protein synthesis, the initiation process can be inhibited, or we can form the toxic proteins, can lead to the misreading of the messenger RNA, forming the toxic proteins or inactive proteins. This is due to the misreading of the messenger RNA. Either we may have initiation inhibition or misreading. Then for tetracycline, so this is the site for aminoglycoside. This site is for aminoglycoside. And ribosomal RNA can inhibit the initiation process. For tetracycline, these ones bind to also at 30S ribosomal subunit at the A site, meaning that we may not have the coming amino acid on the transfer RNA bind to the A site if these ones inhibit this region. Remember, the A site, the P site, they are the interface between the two subunits. But the A site is much in the larger subunit, as you can see. So for tetracycline, they inhibit the process at the A site, but on the smaller subunit here. Those are tetracyclines, meaning that we may not have the formation of the growth of the polypeptide chain will be inhibited. The growing polypeptide chain may be terminated immature. May be terminated. For macrolides, chloramphenicol and nicosamides, these ones, of them, they inhibit the protein synthesis by binding to the 50A subunit. But for macrolides, they bind at the 23S ribosomal RNA near the E site. So we shall have inhibition of the chain here, of the protein synthesis here, around here, near the E site. At the E site. At the 23S ribosomal RNA near the site. Mean that we may not have the bond formation, the peptide bond formation, and we may not have the growing of the chain because there is no exit. For chloramphenicol and glucosamide, also they bind at the 50S subunit at the 23S ribosomal RNA. For chloramphenicol, it binds near the peptidyrotransferase activity. Transferase activity. We say that this enzymatic activity helps in the formation of this peptide bond. Therefore, we may not have the formation of the peptide bond. The chain may be released immature, meaning that these drugs, they are bacteriostatic. So these three classes, they are bacteriostatic. But for aminoglycosides, this one binds irreversibly. These ones bind reversibly, but for it binds irreversibly. And this drug, mainly it is bactericidal. Because we may form the toxic proteins. But these ones, they are mainly bacteriostatic. We just terminate the growing chain. That's why there is no formation of such toxic protein. Thank you for watching. Same the guy. Please don't forget to subscribe, like the video, and share. Thank you so much.